I think we will go ahead and get started today in the interest of time. So it's once again my pleasure to introduce today's Jones Seminar speaker. So today we're hosting Dr. Shrike Zhang, who's an instructor in medicine at the Harvard Medical School, as well as an associate bioengineer at Brigham and Women's Hospital. Shrike got a Bachelor's of Engineering in Biomedical Engineering from Southeast University in Nanjing, China, before crossing the pond over to the United States, where he studied with his PhD advisor, Professor Yanan Xia, at Georgia Tech, earning a PhD in biomedical engineering. And during his time as a graduate student, he did a lot of fascinating work at the interface of nanotechnology, materials science, and regenerative medicine. He subsequently moved to the Northeast, to Boston, where he secured a postdoctoral fellowship position with multiple appointments at Brigham and Women's Hospital, at the Harvard-MIT Division of Health Sciences and Technology, and at the Wyss Institute for Biologically Inspired Engineering at Harvard University. So there he worked with Professor Ali Kadam-Hosseini, working on microfluidics and in vitro tissue culture and in vitro tumor models, so some of the work that I think he'll be telling us about today. So Shrike has had an incredibly prolific career. So I took a look at PubMed and Google just recently. So I think on PubMed, and I don't think these numbers are even up to date, but on PubMed he's got 49 authored or co-authored articles. On Google, that includes more than 80 entries. He's won a number of awards from different organizations, including securing independent funding from the NIH via the K99 Pathway to Independence Award. He was named a Rising Star in Oncology from the Society of Translational Oncology last year. He's one of the recipients of the Young Researcher Awards for the Lush Prize last year. So this is an award that's kind of geared towards inspiring developments on developing in vitro testing alternatives to in vivo models in animals. He's an AAPS postdoctoral fellow awardee. He's earned a fellow award from the Biomedical Engineering Society. He's been named a Baxter Young Investigator. And this is just a list of some of the more recent awards that Shrike has been named for. So with that, I think I will turn the floor over to Shrike, and please join me in welcoming him to Thayer.
Thanks very much, Kyle, for the kind introduction. So I guess the first thing I'm going to do after this is going to go back to my PubMed, and that's the publication back in there. So thanks for inviting me here. So I'm going to talk about our research in the past 3-4 years on developing multi-organ on chip systems for translational medicine and therapeutics. So I'm going to start with the basically the problem in the drug development process. So basically, so this is a like multi-organs on a chip system we have developed over the past 3 years. And then I'm going to talk about how the series of platform technologies that we have innovated over the past few years, including, for example, the microfluidic technologies for emulating the blood vessels, the on-chip biosensing technologies to monitor the behaviors of the organoids over time, and also how the 3D bioprinting technologies have been able to enable us to achieve better like construction of better biomimetic organoids in vitro. So drug development is a lengthy and expensive process. It generally takes about 10 to 15 years and millions to billions of dollars to be able to develop a single drug formulation out of tens to hundreds of candidate drugs in order to pass the strict FDA phase trials. Take the place where I come from as an example. These 3 big pharma companies, Novartis, Pfizer, and Takeda, they together spent more than $25 billion per year just invested into the drug development process. So how is the productivity of the pharma industry? So if we take a look at this graph, over the past 20 years, the total price or cost for developing the drugs have almost tripled over the 20-year period. But if we look at the number of drugs approved every year, it didn't really change, if not reduce, meaning that the productivity of the pharma industry, instead of going up, actually dropped. in the years. And the high failure rate of the candidate drugs have been ascribed to the safety and efficacy issues that have prevented them from entering initially. At the time, those drugs that had already entered into the market were later on also recalled by the FDA due to the toxicities that had not been really discovered during the initial screening process. So the discrepancy between the safety and efficacy observed in non-human models and the human system has been largely due to the inability of the current system models to be able to predict the human responses, whether they are conventional planar static cell cultures or the animal models. So the cell cultures are planar, and then they don't really recapitulate the complex tissue architecture of the human system, and the animal models are genetic different, so they don't reproduce a lot of the human responses towards different drug molecules. So to this end, the organs-on-chip platforms have been developed in recent years as in vitro models of organs that are mimicking the important aspects of the in vivo counterparts. So when these organ models are made of human cells in the right composition, in the right shape or right architecture, they can be used to probe and accurately predict the human responses towards pharmaceutical compounds, chemicals, or even biological species in a very accurate manner. So far, many different types of the organ-on-chip systems have been developed in various ways, and these individual modules can be further interconnected together in a fashion that the integral system also mimic the compartmentalization and also the physiology of the human system, further enabling the investigations of pharmacodynamics and pharmacokinetics as well. When they're made of cells from patients, they also enable personalized drug screening for individual patients. On the other hand, in the human body, all the biophysical and biochemical cues are tightly controlled. In a sense, every single cell, tissue, or organ is functioning as its own sensor to be able to monitor the microenvironmental parameters. In addition, a series of different medical instruments and medical have also been developed to be able to monitor or measure these different parameters from external. So similarly, when an organ-on-a-chip system is produced, we also want to monitor all these different parameters of the organoids on the chips, ideally in a continuous and non-invasive manner, preferably over extended periods of time. However, the conventional ways, for example, ELISA, is usually performed off-chip. They consume relatively large volumes of the medium, and then since everything is manually done, it's laborious and induces frequent perturbation to the organ-on-chip systems. So in order to streamline this entire process, we have built this multi-organ-on-a-chip system containing not only the biometric organoids hosted in an in-house made desktop incubator, but also the series of the sensing units surrounding it. For example, we have physical sensing units, have the electrochemical sensing units, and we have the microscopy units in there to monitor the behaviors of the organoids over time. And everything is controlled in an automated manner with the valve controllers and also the sensing controllers. So we designed our system to be modular to facilitate long-term operation of this entire system and also convenient replacement of individual organoids or individual modules as needed. So this system is composed of a multi-module platform having a central breadboard for routing the fluids and also the microbioreactors, a physical sensing unit, a biochemical sensing unit, and a bubble trap system to be able to remove bubbles from system. Here shows the picture of the entire system. So The central breadboard is used to direct the fluid flow into different modules. For example, showing this movie where we were continuously perfusing the liquid through this bioreactor module and the physical sensing module while keeping the third module here sampled only at predetermined time points. For example, I think it's going to open here right now in a few seconds. So in this way, we can very tightly control when and where the liquid goes into different modules in a very well-controlled manner. So now this opens, so the liquid comes into the third module. So only the liquid won't come into the defined module if we don't want them to be there. So it's very tight control. And then we can also very nicely multiply the capacity to a higher complexity. For example, here we have a chip containing more than 30 different channels, and all the fluid handling here is pretty much automated without any human interference. So due to the complexity of the microfluidics we built, we also developed this miniaturized bubble trap so that when there's bubble coming into the system, we can actually trap the bubble within the device and prevent it from going to the downstream, and then also be able to remove the bubbles from the system using a vacuum by applying the vacuum. And then we built this resealable bioreactor of multi-layer structure. So the good thing with bioreactor is that it's resealable, so we can actually open the top part of the device to be able to load the chamber in the center with organoids that we're interested in putting in there. So with this bioreactor, we engineered a few different types of the tissues or organoids in there, so including the liver, the heart, and vasculature in different forms that I'm going to talk about in the next few slides. So the first one is the liver. So we engineered this functional human liver organoids in the spheroid forms using a mixture of 4 different cell types, roughly in the same ratios as they appear in the human liver. So for example, we have about 80% of hepatocytes, 5% of stellate cells, 10% of the Kupffer cells, and the rest 5% is about endothelial cells. And then we mix them together, and made them into spheroid forms. And then we did show that the liver spheroids actually were functional. They showed high expression of the liver biomarker, including, for example, the tight junction proteins, E-cadherin and connexin 32, and also the cytochromes, for example, the CYP450 and the CYP34A. And then when we loaded this liver organoids into our bioreactors, we could maintain a long-term viability of the cells of over 30 days tested. And more importantly, we then wondered if these organoids were functional or were they actually responding to the drugs. So we did test 2 drug molecules on these liver organoids. So both of them were initially in the market, but were later on recalled by the FDA due to their severe liver toxicity. So we did show that these 2 drugs induced both dose and time-dependent toxicity towards our fabricated liver organoids, showing the functionality of these organoids to be able to respond to the drugs and reproduce human reactions. And similarly, we have engineered functional human cardiac organoids using the spheroid method as well. So in this case, we used a mixture of basically iPS-derived cardiomyocytes, cardiac cells with a mixed population of ventricular and atrial cardiomyocytes, nodal cells, and the cardiac fibroblasts. And then we did show that the spheroids were functional. They had expression of the cardiac-specific biomarker troponin T, alpha-actinin, and also the growth factor VEGF expressed. And also when we put these little spheroids into the bioreactor, they could live as long as 35 days tested, showing long-term viability. And of course, these little spheroids were actually beating spontaneously and synchronously over time. So the beating was uniform and then seemingly propagating from the initiating nodal cells towards the rest of the regions. Similarly, we also did drug testing on these organoids. So for example, we selected 3 different drug molecules that were initially also on the market, but then were later on recalled by the FDA due to the severe cardiotoxicity observed in the human patients. And then when we administered these drugs into the cardiac organoid systems, we did see also dose and time-dependent responses of the organoids towards different drugs, and also the decrease in the beating rate of the cardiac organoids. So we went a little further for making the organoids. So the spheroids are good, but then they're not really mimicking the structures of their in vivo counterparts. So then we used this like photo patterning technology to be able to make human liver and cardiac organoids that are better mimicking their in vivo structures. For example, here's how like liver looks like in vivo. So it has all these little lobule structures in the lobes. So using a photo mask, we were able to pattern the hydrogel loaded with cells into these little lobule structures, and then we did show that we could successfully recapitulate this liver lobule structure with the little cell-loaded hydrogel within the bioreactors, and the cells were also showing high viability over I think it's a 7-day culture period. For the cardiomyocytes, we were also able to reproduce their structures in vitro using the photo patterning technology. So in the human body, cardiomyocytes are aligned into cardiac bundles to be able to exert the stress, the beating of the entire heart. So what we did here is that we actually patterned the surface with line patterns first, line patterns of hydrogels first, and then we seeded cardiomyocytes on top. And then over time, the cardiomyocytes could actually attach and align along these grooves and ridges, and then become eventually beating spontaneously and synchronously over time. And then it did show the strong expression of cardiac biomarkers and also uniform beating rates of about 40 to 80 micro beats per minute, roughly mimicking the human heartbeat. We further, using our bioreactor, we further constructed different vascular organoids. So the first form is a simpler form where we had a multi-layer bioreactor where there's a middle layer of porous membrane seeded with a layer of endothelial cells. So once the endothelial cells become confluent, they could function as a vascular barrier as they do in the human body. So here shows different standings for the biomarkers of endothelial cells. We do show the strong expression of this nice intercellular barrier proteins between the different cells, and cells were, of course, very viable after a few days of culture. So in order to demonstrate the functionality of the vascular organoids, we did a dye diffusion assay. What we did is that we actually loaded the dyes into the bottom chamber, and then we look at how the diffusion of the dye is through the vascular barrier towards the top vascular channel. And then what we showed is that when there's non-confluent endothelial cells, of course, the dye could diffuse very fast in the initial period of time. But then when there was a confluent layer of endothelial cells, we were able to maintain the diffusion of the dye at very low speed until when we infuse this drug, histamine, which is a known vasodilator to dilate the junctions between the cells. So once we inject this molecule into the entire system and dilate the junction between the cells, we did see a fast increase of the diffusion of the dye through the endothelial barrier from the bottom chamber. And then we did show that this vascular organoid is functional, can function as a barrier to slow down the diffusion, but then can also respond to a vasodilator and then become dilated in this device. We can also mimic the biomechanical cues that's happening in the human vasculature. For example, in the human vasculature, there's always a cyclic dilation for the for the vessels due to the pulsing of the heart, pulsing of the blood from the heart. So we could also recapitulate this process on our chips. So what we did is that we had also a 3-layer structure, but for the bottom chamber, we actually applied vacuum. So when the vacuum is applied, the membrane actually is stretched towards the bottom. And then in this case, we can actually very nicely apply the cyclic stretching of the membrane and then also apply the stress to the cells seated on top of the membrane. And then by controlling the amount of vacuum in the bottom chamber, we're able to apply different strain levels to the membrane and also the cells. So, for example, when there's no vacuum, apparently zero strain, and then when there was about 10 kilopascals of vacuum, it's about 9% strain, which is a physiological level of strain. When we further increase the vacuum to 20 kilopascals, this becomes about 16% of strain, which correlates to a pathological level of strain in the human body. So then we showed that with the stretching of the membrane, the smooth muscle cells growing on the membranes were actually responding to the different strands by aligning perpendicularly to the direction of the strand. And then what we found more interesting is that the different strand levels actually induced different different hypertensive phenotypes of the smooth muscle cells growing on these chips. So this black line in the center is the baseline for the smooth muscle cells without any treatment. And then with the physiological strain level, we don't really see much difference in the phenotype of the smooth muscle cells. But what's interesting is that when we applied a pathological strain, we did show that inflammation marker became upregulated in the smooth muscle cells, indicating that the strain, the biomechanical strain, can actually induce cells to actually go into a more hypertensive phenotype and then become inflamed. So going back to the original design that I showed you, so one of the issues that we observed is that there are many tubes connecting different modules. So one of the issues we actually observed for these tubes is that these tubes are pretty much dead volumes in there. So they're inert, and then there's nothing, just medium going on there. So it's actually becoming a lot of dead volumes in between different modules. So one of the ways that we have thought about to solve this issue is to make these tubes actually functional by coating a layer of endothelial cells in the tubes. So when there's medium flowing through the tubes, again, it's not really inert tubes anymore. It's becoming a real vascular-like structure. So we did this by fabricating a series of different PDMS tubes of different sizes to emulate the different regions of the vascular, blood vessels in the human body. And then we could coat another layer of endothelial cells on the inner surface of these tubes. And then we did show that the cells could proliferate and become confluent at about 7 days of culture. They did express, did show strong expression of the endothelial biomarker CD31, and they were confluent surrounding the tubes. And then we did show that these tubes are actually functional, so they actually responded to different drugs infused into the system. So, for example, here we did show that the biomarker levels secreted by the endothelial cells become reduced as more drugs were introduced into the system, and then the cells also died off over time with different drugs. On the other hand, the nitric oxide secretion was also increased when there was a stimulant of drugs infused into the system. So these vascular models are really functional. So then we have built different types of the organoids. So now we're starting to think, are they actually functional together? So I'm going to give 2 examples here to show their potential interactions. So first example I'm going to give is one drug that affects one organoid but not the other one. So in this case, we had both cardiac organoid and the liver organoid built into a single circulation. And then we infused this drug called acetaminophen, which is liver toxic drug, which has not been shown to affect cardiac modules too much. So when we infused the drug into the module, we did see a dose and time-dependent toxicity of the drug towards the liver module here, indicated by the reduced viability of the cells and also the changes of the biomarker secretion levels over time. And as expected, we didn't really see too much of the changes in either biomarker secretion or the beating of the cardiac organoids in the presence of this drug, confirming the kind of the responses of the human body. So in the second example, which is very interesting to me, we showed that two drugs would interact with two organoids. So again, we had the liver and cardiac module. So what happens here is that for the human heart, when there's a drug called epinephrine, when there's this drug called epinephrine present, it's going to bind to the beta adrenergic receptors on the surface of the cardiomyocytes, and it's going to bring the beating of cardiomyocytes up. And then there's another drug called propranolol, which is going to competitively bind to the same receptors in a higher affinity, so that the epinephrine cannot bind anymore. So this drug can actually bring the beating of the cardiomyocytes down to the base level. So this is our organoid culture results. So with only cardiac cells, we did show that when there's baseline 100% of beating rate, and then whereas epinephrine infused into the system, the beating rate was about 1.5 times higher than the control. And then when there's subsequent propranolol infused into the system, then it could bring the beating completely down to the baseline level. However, this is not what happens in the human body, because we have this component called liver. So in the human body, this propranolol molecule can be metabolized by the liver and then become an inactive form that cannot bind to the same receptors anymore. So once it's metabolized, it cannot really rescue the epinephrine-induced cardiotoxicity anymore. So we did show that we can actually reproduce this process also on our chips. For example, here, when we had both cardiac and liver cell organoids on the same chip, when we did the same experiments with epinephrine first and then the propranolol afterwards, we did show that there's a certain level of metabolization of the propranolol, which only brought the beating rate down to an intermediate level, not all the way down to the baseline, meaning that the liver organoids were actually functional, they could metabolize the drug on the chips. All right, so that concludes the first part. So as I mentioned in the beginning, so entire field of the organs on a chip has been focused on improving the functionality of the organoids, but less focus has been placed on the sensing modules that can be built into the systems to be able to, in real time, monitor the responses of the organoids in situ and in a continuous manner. So we have spent quite some efforts in the past few years trying to build all these different types of sensing units into the organoid systems. For example, we have made electrochemical immunobiosensors based on different mechanisms. We have made enzyme sensors to look at small molecules in the culture medium. We have made drug sensors to look at how the drugs are metabolized. We have made ion sensors to measure the concentration of the ions present in the system. We have also made optical sensors for measuring, for example, the morphology of the organoids in the bioreactors. So we have also made a bunch of different physical sensors to be able to monitor the physical parameters, for example, pH, oxygen, and temperature within the whole microenvironment. So today I'm not going to cover all of the different sensing modules, but I'm going to focus on a few different types, including the electrochemical immunobiosensor, the mini microscope, and physical sensors, and discuss how we were able to integrate these with microfluidic devices and also the microfluidic organ chip systems. So we based our immunobiosensor on this electrochemical impedance spectroscopy. So using a gold electrode, we functionalize the surface of gold with a layer of antibodies through a multi-step process. And then once we functionalize antibody on the surface, these antibodies were able to selectively capture the biomarkers of interest from the medium to enable measurements. So this parameter called electron transfer resistance actually changes upon a binding event. And this change of this RCT value can be correlated to the amount of the antigens bound to the surface, and then so we can use this parameter to actually quantify the amount of antigens in the medium. So on the right shows the EIS graphs after each functionalization process, indicating progressively increasing RCT values over time. So this RCT value here after antigen binding is going to be the key value where we can use to quantify the amount of antigens in the cell culture medium. So one of the issues with this kind of sensor is that it's based on immunocapture, antigen-antibody reactions to sense the antigens. So at some point, the surface is going to saturate with all the antibodies bound by the antigens. So we have further developed a few different strategies to be able to regenerate the entire surface. So the first method is basically where we were able to interact the interactions, disrupt interactions between the antigens and the antibodies on the surface, and then reverse this whole surface back to a state where there was only antibodies on the surface. So in this case, we can actually reverse signal back to almost its baseline level using a treatment of sodium hydroxide under flow condition. In another method, we were able to basically strip all the all the surface molecules away and then revert everything back to the pristine state where there's only gold on the surface, and then we can do this whole process again. So this is done basically by using an acid treatment along with the CV sweep, the current sweep at the bottom of the substrate. And under flow, we can efficiently flush away all the cut molecules from the surface and then be able to regenerate the entire surface into its pristine gold status. At AFM images here did show that initially there was the surface was flat with only about 8 nanometers of the height, and then when there's functionalized with antibodies, the whole surface went to a very high roughness, and then after regeneration, it pretty much reversed back to the original state of bare electrode. And of course, we could show the stability over time over multiple regenerations. And then based on these findings, we designed a microfluidic device accordingly to be able to inject all different solutions into this microelectrodes on demand. So using this whole microfluidic chip, we were able to perform all the different processes, including the functionalization, the capture of the antigens for measurement, as well as the subsequent regeneration when needed in an automated process. So comparing these two different methods, method one is fast, since there's only one disruption here, which is the disruption of the antibody-antigen reactions. And then it can be done for about 10 times until before the antibody gets denatured over time. So the method two is relatively slow. It takes about 2 to 3 hours for each of the generation process, since you have to strip away all the molecules from surface and then refunctionalize everything afterwards. So it can last for about 5 to 10 times, depending on the thickness of the gold electrode as a substrate. However, what's interesting here is that since these two different regenerations are based on different mechanisms, we can potentially combine them together. So if we combine them together, we can potentially do this regeneration for up to 50 to 100 times with a single electrode without the need for changing the whole chip for measurement. So this can be, this is actually pretty interesting, and meaning that the whole chip can be used for a longer period of detection for up to 50 to 100 times of measurements until you have to use a new chip. So using this kind of platform, we were able to monitor a series of different biomarkers secreted from the organoids, including, for example, liver organoids such as albumin, ceruloplasmin, A1AT, transferrin, and GSD-alpha, the cardiac biomarkers, including creatine kinase, troponin I, and also vascular biomarkers such as the VWF. So another sensor we built was the imaging sensor. So what we did is that we took this webcam and then we flipped the lens so that it's becoming a magnifying mechanism instead of the original demagnifying mechanism. And then by controlling the distance between this little lens and the bottom CMOS sensor, we're able to achieve different magnifications as well. And then the good thing with that is really small, like this big, so you can fit many of these imaging sensors at bottom of like many different bioreactors to achieve in situ real-time imaging of the organoid behaviors. For example, here shows the migration of your liver cells in a bioreactor over, I think, 24 hours of time. And then you can also look at the beating of cardiomyocytes in there as well. And then the good thing is it's small and it's really cheap. It's like $10 a piece for the microscope. And then with $10, we're pretty happy with the resolution we could achieve, which is about 1 to 2 micrometers. We have also built a bunch of different physical sensors based on optical absorption. For example, we have built oxygen and pH sensors. The oxygen sensor is based on this oxygen-sensitive dye, where it fluoresces when there's no oxygen, and the fluorescence can be quenched by the oxygen, like when there is presence of oxygen. Then by measuring the fluorescence intensity, we can actually quantify the amount of oxygen in the medium. Similar for the pH sensor. So we based our pH sensing on absorption of phenol red present in the culture medium. So phenol red actually changes the absorption intensity at different pH values, and then by looking at this, we can measure different pH values inside the culture medium. So we then built a integrated system where we had different organoids in the system and also a bunch of different physical and biochemical sensors to measure the parameters. So in this case, we had a normal human heart organoid and a human liver cancer organoid. So we showed that we could monitor the physical parameters of the microenvironment over 24 hours of time. We could see the proliferation of liver cancer cells in the bioreactors over the period, and also the beating of the cardiomyocytes within the cardiac bioreactor. And with the multiplexed immunobiosensor, we're able to sense the amount of biomarkers secreted by the different organoids. For example, when we infuse this anti-cancer drug doxorubicin into the system, we did see a killing effect of this drug on the liver cancer organoid. But at the same time, we also saw the side effect of the doxorubicin on the normal cardiac organoid as well. So this platform can be used as a very neat platform for screening both the drug efficacy on cancer tissues and also at the same time the drug side effects on the normal tissues. All right, so in the last part, I'm going to talk about the 3D bioprinting strategies we have developed over the past years to be able to improve the compositional accuracy of the fabricated organoids. So as we know, the tissues in our human body are highly complex, consisting of hierarchically assembled structures across multiple scales. For example, shown here are myofibers in the muscle, lobules in the liver, nephron in the kidney, and also the islet. In the nature, biomolecules at atomic scales are first assembled into nanoscale fibrils and then into microscale building units, which finally assemble into macroscale tissues and organs that are functional. So the challenges here are how to mimic the complexity of native tissue microarchitectures and also how to reproduce synergistic interactions of multiple cell types. There have been a bunch of different biofabrication strategies developed in the past 10, 20 years, but the emergence of 3D bioprinting is truly phenomenal due to the versatility of this manufacturing technology to be able to construct biomimetic tissues in 3 dimensions in a highly controlled manner. In a typical process, the bioink is extruded from the print head, which along with the movement of the print head and the stage allows for deposition of 3-dimensional structures at high flexibility and high spatial control. And the automation process also potentially allows for large-scale fabrication. So in our case, we developed a series of bioprinting strategies for making better organoids. These different strategies span from, for example, this multi-material printer that can improve the compositional accuracy of the fabricated structures, ways to making vascularized tissues, and also potential extension into fabrication of conductive devices that can be used for making bioactuators and biorobotics too. So in the first form, we developed a multi-material bioprinter by combining digitally tunable pneumatic valves into a single print head. So each of these channels is loaded with a unique bioink that can be individually actuated and extruded from the nozzle to be able to deposit multi-material structures at high fidelity. For example, shown here is a video of the bioprinting where all the way from 1 to 7 different materials were printed continuously. And then using the printer, we're able to print complex patterns of multiple materials. Shown here is the Harvard batch of 4 materials. So as the unique advantage of our multi-material bioprinter with a single nozzle design, it's at least 1 order of magnitude faster than any existing multi-nozzle-based system because of the continuous deposition of the different materials in a single printing session. In comparison, for a conventional multi-nozzle-based platform, it requires frequent physical exchange of the different nozzles. And the time for this physical exchange of different nozzles can be as much as the bioprinting process itself. And this requirement for more time of bioprinting is actually multiplied when the type of material is increased. And then using the multi-material bioprinter, we were also able to print 3-dimensional objects, for example, the blocks of multiple materials, concentric rings, blocks with all 7 different types of materials, and the entire human-like structure containing all the different types of tissues in there, biomimetic tissues in there. And the entire bioprinting processes were continuous and requiring no delays between the switching of different materials as well. All right, so when it comes to the cell bioprinting, the choice over the material and also bioprinting parameters become especially important due to the potential damage to the cells caused by the increased shear stress during the bioprinting process. And research has shown recently that overall a shear stress of less than 5 Pascal is going to be minimally affecting the cell viability. So we developed a composite bioink composed of both gelatin methacrylate to increase bioactivity and also a alginate component to increase the viscosity and also allow for temporary support due to the ability to physically cross-link to be able to physically cross-link in the beginning. So we did show that this bioink was shear thinning and at shear rate of bioprinting, which is usually around 1 to 10 per second here, the cells would experience sufficiently low shear stress to be able to maintain high viabilities during the bioprinting process. And then with this optimized bioink, we were also able to print cellularized multi-material structures using a bioprinter. For example, we could mix different cell types within the bioink and print them in biometric structures mimicking, for example, the heart and kidney using the bioprinter. And then the cells were pretty happy in there. They maintained high viability over a period of 7 days, and then they were proliferating over the period as well. We didn't really see much damage to the cells during the bioprinting process. And then we could also print gradient structures using bioreactor. For example, shown here is concentric structure having concentric rings of different concentrations of bone mineral hydroxyapatite in the substrate. And then we did show that with this gradient in the bone mineral, we were able to induce differential responses of the osteoblasts seated on the surface of this structure. They showed differential initial differential adhesion on the rings of different HAP content, and also they show differential proliferation over time as well. So another focus has been on vascularization. So we all know that vascularization is a very important process, since no single organ in the body does not have a interconnected vascular network. So in a previous strategy, we used the sacrificial bioprinting for making the vascularization patterns in the hydrogels. In this case, we first deposited hydrogel fibers of agars, and then we casted the hydrogel, like the tissue construct surrounding these microfibers. And afterwards, after we cross-link the hydrogel blocks, we could then remove the initial hydrogel microfibers from the blocks to actually make these hollow micro channels mimicking the structure of the blood vessels. And what's interesting is that we can further seed a layer of endothelial cells within these channels to make them also biologically active. And of course, all these channels are hollow, just like the blood vessels in the human body. Not only can we make normal blood vessel structures, we can also make them make models of vascular malformation. For example, here we did a model of thrombosis. So thrombosis is a pretty heavy healthcare burden in the U.S. with an incidence rate of 1 in every 500 Americans. It is caused by the formation of the blood clots in the blood vessels of the patients. And then we did show that with these endothelialized little channels, we were able to also infuse human whole blood into the channels and let them clot within the channels to mimic this thrombosis process. And then with microscopy images, we did show that there's endothelial kind of layer surrounding the surface of the channel, and also we showed there's aggregated red blood cells within the channels mimicking the clots. And then histology images also showed very nice similarity between our model and a real human thrombosis. They're both aggregated red blood cells within the channel, and also we showed lining of the endothelial cells on the side. So I'm going to probably skip this one for now. So another way is we have also developed a way to build hollow vascular structures in a single step. In this case, we actually had a 3-layer print head where the bio ink is extruded from the central layer, where the cross-linking agent calcium chloride is streamed from both the inner layer and also the outer layer. So in this case, when we extrude the bio ink, we were able to directly form the vascular, hollow vascular structure very immediately. And by modulating the parameters of the bioprinting, for example, the sizes of the concentric needles and the flow rates of the different phases, we were able to fabricate the tubular structures of different inner diameters, outer diameters, and also wall thicknesses to model the size of a range of different vessels in the human body. And using bioprinter, of course, we could deposit them in any arbitrary shapes that we want and in 3 dimensions. And of course, these hollow fibers that we printed were perfusible, just like the vascular structures in the human body. We showed very nice perfusion of the air throughout the entire construct. And then we can also encapsulate vascular cells within the structures. And then we showed the development of the confluent zooming structures within the hollow, within the walls of the hollow tubes to mimic the biological functions of the blood vessels. And then using these bioprinted little vascular beds, we were able to further develop vascularized tissues. For example, in this case, we were able to further see the secondary cell type into the interstitial space of the fibers, and then be able to construct biomimetic and cellularized myocardium that's mimicking their in vivo structures. So here shows the images and movies of the cardiac organoids we built there. So the confocal images showed the lines endothelial lining in the fibers and the presence of the cardiomyocytes on the surface of the vascular bed. And then, of course, the cardiomyocytes were also spontaneously beating over time in the bioreactors. And then by modulating the bioprinting parameters, we can also modulate the microarchitecture of the scaffolds we print. For example, by extending the unit aspect ratio of the unit grace from 2:2 to all the way to 2:5, from like anisotropic to isotropic, we were able to modulate the organization of the cardiomyocytes on top from a random organization to a more aligned organization to better mimic the cardiac bundles in the heart. And then, well, of course, we also, besides the expression, the structural organization of the cardiomyocytes and also expression of the cardiac biomarkers, we also showed a much improved beating of the cardiac organoids over time when they were cultured on a scaffold with higher aspect ratio and better alignment. So we know that cardiomyocytes are communicating in the human body to be able to transduce the signals, electrical signals between the cells for achieving the uniform beating across larger area. So we then thought that if we can actually introduce conductivity into our bioink, we were probably able to improve the functions of the cardiomyocytes within these hydrogels. So we did show that it's working. So we actually mixed these gold nanorods into our hydrogel bioink, and then we did show that when the cardiomyocytes were cultured on top of this, these are the bottom row is showing the pictures of cardiomyocytes growing on the conductive bioinks, and then we did show that the cells were growing much better. They had much better spreading and expression of the cardiac biomarkers when they were cultured on the conductive bioink compared to when they were cultured on the normal non-conductive bioinks. And then the beating, of course, were much improved on the conductive bioinks due to the better signal transduction between the cells as compared to when they were cultured on the substrate which is non-conductive. And then we could also mix these conductive bioinks with cardiomyocytes and then use them as the conductive bioink for bioprinting. And then we did show that with bioprinting, we were also able to improve the spreading and the contraction of the printed cardiac tissues with the conductive bioink compared to when there was no conductivity within the hydrogels. All right, so we also made another conductive bioink, which is even more conductive. So in this case, we actually mixed GM, this gelatin methacrylate molecule, with DNA and CNTs, carbon nanotubes, to be able to form a network of carbon nanotubes within the hydrogel so that the entire bioink is very conductive. And then using bioprinter, we can actually print this conductive bioink on any substrate, including those that are flexible. And then since there's presence of this GM component, which is bioactive, we could also culture the cells on top of this printed bioinks to showing the biocompatibility of the conductive bioink. And then with this conductor bioink, we were able to also fabricate the bioactuators out of it. So for example, we were able to show that using the multi-material bioprinter, we could print a series of different bioinks with different concentrations of carbon nanotubes inside. Therefore, the different conductivity of each line of this circuit. And then in a parallel circuit, we did show that with different resistance in each line, there was a difference in the luminescence of the LEDs connected in each of the lines over there. We could also print the whole entire circuit in 3 dimensions within a hydrogel, potentially serving as intrinsic electrodes for measuring signals. And then we could also potentially design bioactuators using the bioprinter where we can align cardiomyocytes on top of these printed conductive devices to be able to direct them into the right shape for actuation. For example, here are some examples showing that we can actually print structures mimicking a stingray, and then we can subsequently seed cardiomyocytes on top to in order for them to spontaneously beat or contract in the culture medium. All right, so with this, I want to summarize with research. So basically, we have built a multi-organ on a chip system containing a multitude of sensing units in there, including electrochemical biosensors and also the physical sensing units to be able to monitor the biochemical and biophysical parameters in real time in a continuous manner over extended periods of time. And using the microfluidics, we're able to measure we were able to emulate the vascularization or like interconnections between the different organoids within the integrated system. And then using bioprinting, we're able to also improve our design for the organoids to be able to better mimic their in vivo structures for better functionality. All right, so I'm going to acknowledge the whole, I think this is the entire lab, and then we have about 90 people in the entire lab with a few different kind of PIs in there. And I would also like to acknowledge all the funding agencies and also all the collaborators, and I would like to thank everyone for coming here, and I can take any questions.